Hello, my name is Bruna, and I'm a second-year PhD student in the Plant Insect and Microbial Sciences program at the University of Missouri. My advisor is Dr. Gary Stacy, and my research is aimed at describing new genes involved in the soybean-Rhizobium interaction. As a brief introduction, legumes such as soybean are able to interact with rhizobia, nitrogen-fixing bacteria. The bacteria are housed in root nodules, where they fix nitrogen and provide nitrogen to the plant. This is particularly important in agriculture because it reduces the need for nitrogen fertilizer, which is both expensive and polluting. Understanding the molecular mechanisms of this interaction is important to be able to transfer nitrogen fixing capabilities to other crop plants, such as maize, which would make agriculture more sustainable. With this in mind, we used translating ribosome affinity purification, or TRAP, to find new genes involved in this interaction. We used a cortex-specific promoter to drive the expression of flag-tab ribosomal proteins. In this way, we are able to purify RNA that is being translated only in the cortex. We use this with soybean root samples that were inoculated with Bradyrhizobium japonicum or mock inoculated. So samples were taken at 48, 72 hours, or 96 hours after inoculation, and then RNA-seq was performed. We described a few upregulated genes, and we decided to focus on 80 genes that were both upregulated both at the 72 and 96 time point. With these 80 genes, we used them further for characterization via RNAi knockdown. So soybean transgenic roots with RNAi constructs were made, and these plants were inoculated with Bradyrhizobium japonicum. 4 weeks after inoculation, assessed nodule phenotype. In this case here, I'm showing the number of nodules per root, and this is our empty vector control. We have examples of 3 genes here whose nodule numbers were affected. The first one that I'm showing here, GA3BDO, encodes for gibberellin dioxygenase, which is involved in gibberellin biosynthesis. Hormonal regulation of nodulation is complex, and gibberellin is known to play different roles in early and late stages of nodulation. These 2 other genes that are involved in nodulation and when knocked down lead to higher number of nodules are both transcription factors. And WRKY and MIB transcription factors are involved in different biological processes, but these specific transcription factors haven't been studied specifically in relation to nodulation. So the next steps would be to further investigate how the spatial and temporal activation or expression of these genes affects nodulation. Thank you so much. I'm open to any questions, and feel free to contact me. Here's my email and my lab website. Thank you.